So I'm gonna start the recording now. Yesterday during one of the labs, I forgot to press record, but it wasn't a long lab overview, so it didn't affect them. I remembered this morning. I've got post-it notes everywhere. Okay, everybody, welcome to Chapter 11, Cell Communication, or as I call it, Intercellular Communication. I want to stress, unlike photosynthesis, where you might be able to get by with my notes that I posted, the PowerPoint I posted in the review, this Chapter 11, it's heavy into what we call advanced cell molecular biology and physiology. Okay, here at ACC, they cover this usually in AMP2. And by then, students are supposed to have a very strong background between 1406, maybe 07, microbiology and AMP1. So make sure that you also, in addition to using your notes and the PowerPoint review that I posted, make sure you read the text and just read what I cover in the notes. Were y'all able to hear what I said in the beginning here? Because again, I for some reason keep forgetting to put the mic closer. All right, the advantages to being loud. I remember when I used to be very soft-spoken, believe it or not. Okay, thank you, God, I am loud. All right, louder, I should say. Okay, let's go ahead and get moving here. So, thank you, Noah. Okay, so if you look there, the second slide is common properties of organisms. Well, we talked about 7 common properties or traits that living organisms must possess. In other words, when something is analyzed by scientists and they want to deem if it's living or not, it has to possess these 7 traits to consider it living. Okay, well, when it comes to cell communication, okay, what I've highlighted is areas where cell communication plays a role. But technically, I could highlight the whole thing. Okay, but what I've highlighted is in order for our bodies to respond to our surrounding environment, that requires the cells of our body to be able to communicate with each other. Okay, reproduction. Okay, you might have heard of the endocrine system and the reproductive system. That is a lot of cell communication that is going on, controlling development. Well, just like when infant's born and an infant develops into a toddler or a child reaches puberty, okay, that's a lot of cell communication that's going on that's resulting in changes. Energy utilization. When you exercise, cells of your body communicate with each other. It can result in everything from additional chemical messengers being produced that tell your skeletal muscles, hey, ramp up the rate at which you're producing ATP because we need you to produce enough. Or it could be simply your nervous system telling your cardiovascular system, hey, you need to increase that heart rate. Or telling your respiratory system, you need to increase that breathing rate. And then regulation. Cell communication is the key means, okay, for regulatory mechanisms in our body. Okay, your nervous system and your endocrine systems, 2 of 11 organ systems in our body, okay, they use cell communication to regulate body functions. So this is a heavy area here that we're talking about as far as cell communication goes. It's a key area. I'm sorry, I said heavy, but key. All right. The 2 stars to the show when we talk about cells communicating with each other are molecules called ligands. In lay term, ligands are known as signaling molecules, which is right here, just to the left. Okay. The other star of the show, these are proteins. Okay, we don't just say molecules, but they're proteins that we call receptors. If you recall, we talked about in the membrane of cells are transmembrane proteins, and these transmembrane proteins have different functions. They could be CAMs, or they could be involved in membrane transport, or they could function as receptors. Okay, receptors are proteins. Now the way it works is for ligands. Okay, they're signaling molecules, but they're better known as hormones and neurotransmitters. This is the abbreviation for neurotransmitters. Okay, I give you reasons for why do we call some of these chemical signals or signaling molecules ligands, hormones. Well, any chemical signal that's a ligand that's released into your blood, that's a hormone. Any chemical signal that's a ligand that's produced by neurons of your nervous system in order for those neurons to communicate and control other cells, okay, we call these neurotransmitters. So if you ever hear the terms hormones and neurotransmitters, they're talking about ligands. And simply put, ligands are signaling molecules. Receptors. Well, if you look up here to the left at the very top corner, okay, they're showing you 2 neighboring cells. The purple area represents the nucleus of each cell. They take a piece of each cell, and this cell above is the signaling cell. In other words, it's the cell that has something to say. And what it says, it's going to say to a cell that might be nearby or far away. But in either case, the cell that is going to do the listening and respond to what is told, that is known as a target cell. It is in the target cell that you will find the second major compound called a receptor. Okay. Again, receptors are proteins. Many are transmembrane proteins, as I put here. However, if you look down here in the bottom right corner, some protein receptors are what we call intracellular receptors. Sometimes you'll hear them referred to as cytosolic or nuclear receptors. Okay. Now, the reason why you can find these proteins that function as receptors, either in the membrane or within the cell, okay, has to do with the chemical structure of the ligand, which we'll go over. Some ligands are not lipid soluble, therefore they cannot diffuse through this phospholipid bilayer. Some ligands are lipid soluble, therefore to reach their target cell, they'll actually pass through the membrane and then bind to the receptor. So transmembrane proteins that function as receptors, if they're in the membrane, we actually call them cell surface receptors. But if the receptors are always located inside of the cell, we call them intracellular. I think everybody's heard of the fight or flight mechanism. Okay, well, fight or flight mechanism, that definitely involves cell-to-cell communication. All right, your nervous system and endocrine systems are basically telling every other system of your body, including within themselves, we need to ramp it up. Now, it could be, hey, we need to increase the level of activity, okay, because we're hungry, or we need to increase the level of activity because we don't feel like being on the dinner menu. So if you look here, fight or flight response in the top. Fight or flight response is triggered by a signaling molecule called epinephrine. So epinephrine's a ligand. You'll hear talk if you ever take, like I said, a physiology course or an endocrinology course, you'll find that epinephrine can be a neurotransmitter used by the nervous system, or it could be a hormone used by the endocrine system. Okay, you may have heard of EpiPens. Okay, most people know this, but the epi comes from epinephrine. Epinephrine is more popularly known as adrenaline. Well, it just so happens that target cells that have receptors that recognize and bind to epinephrine, the name of those receptors on target cells is adrenergic receptors. This is a general class of receptors, adrenergic receptors. In advanced classes, they'll talk to you how there are different forms of this receptor found on different cells. But what I want you to know is the general name for a class of receptors called adrenergic receptors that are found on target cells that bind to the ligand epinephrine. And that's what it's showing you down here. It's showing you the molecule epinephrine and how it's binding to its receptor. Okay? Now, the way it works is that sometimes when a ligand's released, okay, it's a hormone. In other words, it's a chemical signal released into your blood. So it's going to pass by a lot of cells. Okay? Sometimes when the ligand's released, the target cell is right next door, but some of that ligand, that signaling molecule, might diffuse away or in the wrong direction. So to ensure that signaling molecule, okay, stimulates its target cell, in other words, regulates its target cell, the ligands will bind only to specific receptors like a locking key mechanism. So going back to the previous slide, I'm sure all of you, like me, you have a variety of keys, a lot of keys on a keychain, but you know those keys don't all unlock the same door or start the same car. Okay, the keys have to have a certain curvature or a certain shape to them to unlock certain locks. So if you look here back on the previous slide, what I'm circling with the little hand, okay, all receptors, it doesn't matter whether they're cell surface receptors or intracellular receptors, all receptors have a region in them called a ligand binding site. That ligand binding site of the receptor has a shape that only complements the shape of the ligand or signaling molecule it's supposed to bind. This is what I mean by locking key. The lock is a receptor, the key is the signaling molecule called the ligand. So the shapes have to complement each other, okay? If not, If the ligand shape doesn't complement the ligand binding site shape of the receptor, then it's like nothing's there. So the target cell, which is over here to your left, it's just standing there like, hey, I don't recognize anything. Here it's showing you at the top right corner. The cell releasing the ligand is doing the talking. That's one way to think about it. And the cell with the specific receptor, the target cell, it's doing the listening. So over here, one cell is doing the talking, and the one with the receptor is doing the listening. Sometimes the cell doing the talking, the signaling cell, okay, when it does the talking, in a manner of speaking, it has to shout. In other words, it has to release its ligand into your bloodstream because the target cell is far away. That's like you shouting loud as you can to reach somebody who's off in the distance. Sometimes the signaling cell, the person doing the talking, is in your ear. In other words, sometimes the cell doing the talking is right there, up against, but not quite touching. It looks like they're touching here, but what they've done is they've taken this area right here between these two neurons, and they're trying to show you there's a little bit of space in between these two guys. If you have a job, you may have had this happen, or you may have done this if you're a supervisor. You get up right up in the ear of somebody, and you make sure they hear you loud and clear. So hopefully you haven't. Hopefully you haven't had it happen to you, although I am a loud talker. All right, so now if you look at the next slide, okay, this cell-to-cell communication, this allows for the regulation of various cellular functions. An example of a type of function that's regulated, well, it's simply the production of blood cells, okay? Blood cells, that does not just mean red blood cells. Blood cells means white blood cells, red blood cells, or what we call clotting factors called platelets, which come from cells. So if you look here, I believe everybody's heard of bone marrow. In adults, one of the key areas that you will find the type of solution called bone marrow, okay, is in our pelvic or hip bones. You may have heard about bone marrow transplants. Well, what it is with the bone marrow transplant is that the bone marrow, this solution-like substance, contains what we call stem cells, okay, more specifically adult stem cells, okay. What we mean by stem cells is stem cells that have not fully matured. They have not fully differentiated into a specific type of cell to perform a specific type of function. However, in this case, in your red bone marrow, these stem cells called hematopoietic stem cells, they're adult stem cells because they have partially differentiated, or they have matured some to where they are already committed to becoming a blood cell. It's just we don't know what type of blood cell. Now, the way this works in regards to cell communication, if you look here to the right where it says erythrocytes, that's a technical term for red blood cells, okay? If you go a little to the left and just above the arrow, there's an abbreviation, EPO. Usually it's all three in uppercase letters, EPO. EPO stands for erythropoietin. This is a ligand, or a hormone, as they more specifically say, produced by cells of the kidneys. Cells of the kidneys can monitor O2 levels, and if they detect O2 levels are a little low, they can release this ligand or hormone called erythropoietin into the blood. Erythropoietin will enter the blood and make its way to wherever there is bone marrow that contains these stem cells, okay? These stem cells, such as hematopoietic stem cells, they're not showing it to you, but they have receptors with a ligand binding site that complements the shape of EPO. Once those receptors bind EPO, what will happen is a stem cell, okay, will respond, okay, to that chemical signal by differentiating or maturing till eventually it completely changes into a red blood cell. It'll continuously receive this message, though, over and over to make sure that it knows, hey, like breadcrumbs, keep following me this way till the final response, it's a mature red blood cell. Now it's differentiated into a specific blood cell type to perform a specific function. So if you notice the EPO here is in a purple color, that means all these others that are listed here, all these other abbreviations, these are abbreviations for other ligands, okay, that will target the stem cells in your red bone marrow, telling them what type of blood cell to specifically turn into, to mature into, okay? Now, the way cells communicate, well, it varies. Sometimes cells communicate through their cytosol. We talked about CAMs that can form gap junctions or implants, plasmodesmatas, sorry about that, tongue-tied, okay, to where two cells are connected to each other, okay, with the help of proteins. But in addition to that, when their cell membranes connect to each other or cell walls, there's a little tunnel or portal or pore that's created that allows them to directly communicate with each other. But they're still using signaling molecules called ligands. Sometimes two cells will communicate with each other through direct contact. One of the cells, the signaling cell, it will not release the ligand into the ECF. Instead, it'll stick it out of its membrane and just literally walk up to the target cell and physically insert the ligand into the receptor. Notice for this receptor, it has a curved ligand binding site that perfectly complements the rounded shape of the end of the ligand in green. Okay, so your lock and key mechanism. So it basically says here, I'm going to put the ligand into your receptor, and then this cell, target cell, will carry out a response. Sometimes the cells are not in direct contact with each other, but they're close to each other to where now the signaling cell can release its ligands into the ECF that is adjacent to the target cell to ensure that the target cell always gets the response, that always gets the signal and responds quickly. Okay, you see this for the nervous system. This is what I mean by a supervisor standing near you and your ears right there by their mouth, and they're making sure you hear everything they tell you, and then you have to respond to it. But sometimes, and this is the case of the endocrine system, sometimes the signaling cell, okay, is located distal or far away from the target cell that it needs to regulate or communicate with. In that case, these signaling cells, instead of being called neurons like over here, we often call them endocrine cells. They release ligands, more popularly known as hormones, into the blood. So as they're showing you here, okay, if the signaling cell is located here in the upper torso, but its target cells are located down in the pelvic region, the best way for those chemical signals called ligands or hormones to reach the target cell is through your blood. Eventually, they'll diffuse out of your blood and reach the receptor on the target cell. Now, when this happens, these ligands are actually coming out of the blood long before they reach their target. Okay, but not all of them, but some of them. So they're coming in contact with other cells. However, only the target cell will have a receptor that recognizes the ligand. So that ensures, okay, that only the target cell gets the response. It's kind of like you standing in a crowd and somebody calls out, but they call out your name. Okay, or you're in a crowd and they call out somebody else's name. That's going to determine whether you pay attention or not. Okay, next part has to do with type of ligands based on, I put structure, but what we're talking about is chemical structure. Scientists, when they discovered these signaling molecules called ligands, okay, wanted to uncover their shape or structure. One of the reasons for doing that, well, hey, if we uncover their shape or structure, that might actually help us, okay, figure out what the receptor will look like and where to look for the receptor. Will it be cell surface or inside? The other reason, well, that has to do with pharmaceuticals. Can we create a synthetic form of this molecule, this ligand, okay, so that we can give it to somebody for medical purposes? So if you look here at ligands based on their chemical structure, or if you want to call it their anatomy or shape, some ligands are said to have a chemical structure referred to as an amino acid derivative. They're not amino acids, but they come from amino acids, but they do not have the same function as an amino acid, which we'll talk about in a bit. Some ligands have a chemical structure, okay, that actually looks like a very, very tiny protein. Well, in biochem, very tiny proteins are called peptides. So if you ever take a biochem course and your biochem professor starts mentioning peptides, automatically tell yourself, well, he's talking about proteins, but he's talking about very tiny proteins. Proteins that are formed by maybe as few as 9 amino acids up to 100 amino acids covalently bonded, such as insulin. So what they call these are peptide hormones. Okay, so this is another type of ligand based on its chemical structure because it's a really tiny protein called a peptide. They call them peptide hormones. Okay, so remember, if I ask you to list the names of ligands based on the chemical structure, you can't just say peptide because not all peptides function as ligands. Only those called peptide hormones. And it's going to be the same for this third type. Based on the chemical structure of ligands, some ligands were found to have shapes similar to steroids. Okay, well, now they're classified as steroids. However, not all steroids function as ligands, but some steroids do. Well, what they found is some ligands have a chemical structure, okay, very similar to a steroid called cholesterol, but cholesterol is not a ligand, but these are ligands coming from it. So they're steroids, but since they function as ligands, unlike cholesterol, they call them steroid hormones. Okay, so I'm going to go back here. Don't forget, if you have a question, just ask in the chat box here. Now, for the first type of chemical structure of ligands, okay, they call them amino acid derivatives. A key type of cell that you'll find these types of ligands being produced are just neurons of the nervous system, okay? If you look here, some signaling cells convert an amino acid into an amino acid derivative, and the amino acid derivative functions as a ligand. An example of an amino acid that is structurally altered to turn into an amino acid derivative or a ligand is tyrosine. If you recall, tyrosine was one of 7 amino acids you had to know. There are neurons in the nervous system that contain enzymes that have the ability to take tyrosine and change its chemical structure, turning it into the amino acid derivative dopamine. I forgot to put the parentheses on both sides. It's over here. The abbreviation for dopamine is DA, so just add in an extra parenthesis. Okay? So dopamine is a ligand, and based on its chemical structure, it's a ligand also called an amino acid derivative. But dopamine is not one of the 20 amino acids. Okay? Now, some neurons have additional enzymes in them that allow them to take dopamine and chemically alter its structure, transforming it into norepinephrine. Norepinephrine I refer to as a cousin or sibling of epinephrine, and it's over here. The abbreviation for norepinephrine is NE. You might have heard the term noradrenaline. This is what they're referring to. And then some neurons have additional enzymes that can take this amino acid derivative, this ligand amino acid derivative, norepinephrine, and convert it to adrenaline, better known among the scientific community as epinephrine or epi. Okay, so you can see here, these are 3 examples of ligands that based on their chemical structure, they're called amino acid derivatives, but they function as ligands. All right, they come from amino acids, but they are not amino acids. All right, now the other importance to knowing their chemical structure that might allow a pharmaceutical company to come up with a synthetic form, okay, create one comparable, is it also allows scientists to know where to start looking for a receptor on potential target cells. Amino acid derivatives are not lipid soluble. What I mean by that is they cannot freely diffuse across the plasma membrane. They can't get past that phospholipid bilayer. So by uncovering the chemical structure of these types of ligands called amino acid derivatives, that told scientists, hey, on those cells that I believe might be potential target cells for any one of these amino acid derivatives, I want to start searching for the receptor in the membrane of the protein. In other words, I want to look for transmembrane proteins that function as cell surface receptors. Okay, so this is where the importance comes in. Peptide hormones. Okay, I said a peptide is a very tiny protein, but not all peptides are ligands. So for only those peptides that function as ligands, we call them peptide hormones. All right. Some signaling cells carry genetic information for how to make a peptide hormone that functions as a ligand. Listen carefully. I said a peptide is a type of protein, but the way it works, and we're going to talk about this more as the semester goes, is I said proteins do the work in the cell, but what I haven't talked about, I believe, or talked about much, is how does a cell know how to make that protein that it needs to do work? Well, a cell can find the recipe on how to make a specific protein to do a specific type of work in its DNA. We'll talk later how regions of DNA form units called genes. And all the genes you inherit in your DNA make up your genetic information or genotype. then you're going to hear me say that each gene serves as a protein recipe, tells the cell how to make a protein. Now, the reason why I'm talking about this, well, a company called Genentech, back around the 70s, early 80s, okay, they were studying type 1 diabetes and insulin. And they knew for type 1 diabetics, the only cells that could produce insulin located in their pancreas, they were slowly losing them. Okay, so as they lost the only cells that could produce this peptide hormone, this type of ligand peptide hormone called insulin, what they did is, hey, with the help of biochemists, we uncover the structure of this hormone insulin. When they realized the hormone insulin is a peptide hormone, a tiny protein made up of amino acids, they start looking into the genetic information of human beings to locate the gene or protein recipe that tells the cell how to make insulin. Once they identified that gene and isolated that gene, okay, they were able to create GMOs that would produce large quantities of human insulin, which would then be purified and sent out to the medical community to distribute among those type 1 diabetics. It was a huge advancement. So I'm just giving you reasons for why it's important to know the chemical structure and why it's relevant for us to talk about it in this class. Other examples of peptide hormones, okay, tiny proteins that function as ligands, are oxytocin and vasopressin. Oxytocin, it's a ligand or a peptide hormone most popularly associated with labor contractions when a woman is giving birth. And vasopressin, okay, it now more popularly goes by its alternative name, ADH or Antidiuretic Hormone. This peptide hormone has multiple functions, and when you take an A&P class, they will talk about it in terms of water balance and regulation of blood pressure. Okay, so it's got dual functions. But if you look at all three examples I've given you, okay, you can see the three-letter abbreviations, some of which you were responsible for, such as CYS, TYR, or GLY. The reason for that earlier in the semester is so you have an idea of what you're looking at in these figures. So what they're showing you are amino acids, and the lines indicate covalently bonded to each other. To try to explain what I mean up here, ligands that are peptide hormones are tiny proteins. Now, what's the other advantage to knowing the chemical structure of these types of ligands known as peptide hormones? Well, they're not lipid soluble. So this let the scientific community know that any cell that they felt was potentially a target cell of any peptide hormone, they knew, hey, they're not lipid soluble. They can't freely pass across the membrane, so we'll search for the receptors beginning in the cell membrane, the plasma membrane. In other words, we will search for cell surface receptors, okay? The last type, based on chemical structures, are ligands called steroid hormones, okay? Remember, steroids are one of 4 types of lipids, but you have to remember, not all steroids are ligands, okay? Only certain types of steroids function as ligands, and I have them down at the bottom. 3 examples, I have them also up here: testosterone, estrogen, and cortisol. Okay. All 3 of these ligands are derived or come from the precursor cholesterol, which is above. It says some signaling cells contain enzymes that convert cholesterol, a precursor. In other words, what we mean by precursor is the starter material that is going to be transformed into a steroid hormone. Okay? Cholesterol is a precursor to steroid hormones. Steroid hormones function as ligands. So again, if I ask you to list the 3 types of ligands based on chemical structures, don't give me amino acids, give me amino acid derivatives. Don't give me peptide, you have to give me peptide hormone. Same thing with steroid, don't give me steroid, give me steroid hormone. You have to learn to be technical when you go into the sciences because there is a difference. So this figure down here is explaining what I mean by these 2 lines here. Cholesterol is a precursor and 3 types of steroid hormones. In the testes of males, there are cells that can take this steroid called cholesterol. I'm sorry, Javdi, did you have a question? Just ask me on the chat like you did last time if you can. If not, re-raise your hand. Okay, so down here, okay, cells in the testes of the male, okay, they contain, when they take up cholesterol, they have enzymes that can tweak the chemical structure of cholesterol, transforming it into a ligand, or more specifically, the steroid hormone testosterone. In women, in cells of the ovaries, there are cells that can take the steroid cholesterol. And there are enzymes in those cells that can tweak the chemical structure of cholesterol, transforming it into estrogen. Okay? Cholesterol is only a steroid. It's not a steroid hormone. However, estrogen is a ligand known as a steroid hormone. And then in the adrenal glands, if you're not familiar with the adrenal glands are located on top of the kidneys. Okay? So what will happen, sorry everybody, I get scam calls a lot. I don't know about y'all. It's gotten real bad lately, but back on track here. Yes, I do want you to know examples of each type of ligand for the exam, such as estrogen. Yes, exactly. Everything I'm covering, Jobdi. Did everybody else hear that? Jobdi asked the question about, do you need to know the examples of each type of ligand I'm giving for each type of chemical structure? Absolutely. Okay. Now, if you look here, for cells that are located in the adrenal gland, which sit on top of the kidneys, those cells have enzymes. Some of those cells have enzymes that can take this steroid cholesterol and transform its chemical structure, change the chemical structure, transforming it into the steroid hormone cortisol. Okay. In A&P, but not in this class, but in A&P, they'll talk about cortisol and its anti-inflammatory properties, but they'll also talk about cortisol in relation to glucose balance or blood glucose levels. Now, keep in mind, okay, even though testosterone and estrogen are ligands, okay, that we refer to as steroid hormones, okay, they all come from cholesterol. They're all steroids. Why am I pointing that out? If you recall, I had pointed out during membrane transport that gases and lipids can readily or freely diffuse across the phospholipid bilayer of the cell membrane. Okay. Well, these three steroid hormones, these three ligands, well, they all come from cholesterol. So they're all still lipid soluble. Therefore, testosterone, estrogen, and cortisol, okay, are lipid soluble and can freely diffuse across the cell membrane of the target cell. Therefore, when scientists discovered the chemical structure of these ligands, okay, and that they were steroids, but specifically steroid hormones, that told them on any potential target cell, they would actually look for the receptor inside of the cell. Well, if it was found to be true. Steroid hormones, the receptors for steroid hormones, they are located inside of the target cell. We call them intracellular receptors. Sometimes they'll more specifically call them cytosolic or nuclear receptors. Now, what we're going to change directions a little bit. Now we're going to focus on the target cell. Okay. There's a three-step process when it comes to cell communication on the target cell side. This three-step process has to do with, okay, the target cell receiving a signal, how that target cell transmit or transduces that signal to the correct area within it so that in the end the target cell can respond. Okay, it's being told to do something and it needs to respond and do it. It's a three-step process. The first step, and I'm going to show you this PowerPoint a few times, so just look at the bottom to know which step we're on. The first step, step one, is called reception. Reception is simply where the ligand, or let me go back to this one, where the ligand or signaling molecule binds to its receptor. In this case, they're showing you a transmembrane protein, or more specifically, a cell surface receptor. The ligand binding side of the cell surface receptor is facing the ECF. And the ligand binding site has to have a shape that complements the shape of the signaling molecule or ligand so that reception can occur. This over here is just showing you that some of these transmembrane proteins, they're actually very large. They're not that simple. But over here, if you look, just look at this side for now. Okay. They're showing you a target cell, and the target cell specifically is a smooth muscle cell. In the membrane of smooth muscle cells, you will find what we call adrenergic receptors. These adrenergic receptors have a ligand binding site that faces the extracellular fluid, and that ligand binding site has a shape that complements the shape, okay, of the amino acid derivative or ligand epinephrine. And they're showing you how they fit like a lock and key. Okay, so once epinephrine binds to its cell surface receptor called an adrenergic receptor, what they're not showing you is that this is going to cause a change in the receptor. We'll talk about that in a second when we talk about step 2. However, receptors behave differently. They all don't behave the same. Okay, when I used to go over the skeletal muscular system with my students in A&P, we used to talk about the neuromuscular junction, and we used to talk about how skeletal muscle cells, they have receptors, but those receptors have a dual function. On one end, their job is to recognize and bind to specific signaling molecules called ligands. But what I mean by dual function is that these receptors also functioned as ligand-gated channels. When they would bind to the ligand that they were specific for, that would cause a change in the shape of the receptor, the cell surface receptor, and it would allow ions to diffuse. Some receptors, okay, they don't have dual functions in regards to functioning as ligand-gated channels, but they do have dual functions in regards to they're not only receptors, but they behave as enzymes. Okay, so what will happen is some of these receptors that are cell surface receptors, their ligand binding site, as shown here, will bind to the signaling molecule. That can cause a change, say, in more than one receptor. And then eventually what happens to the receptor is it starts acting like an enzyme, catalyzing some type of chemical reaction near the cell membrane, but on the ICF side. But most of the time when you hear talk about receptors, and I know they visit this in A&P, I believe it's 2, they'll talk about receptors referred to as G protein coupled receptors. Okay, we'll talk about this in a little bit because the G protein part has to do with an intracellular protein, okay, that can move around the inside of the membrane. Therefore, it can move to a cell surface receptor or it can move away. It all depends on whether or not the receptor has performed the first step, reception. This right here, this slide, is an example of an intracellular receptor. I'm going to use this here to the left. They're showing you out in the ECF, we have our steroid hormone, testosterone. It's a steroid, so it can readily diffuse or easily diffuse through the phospholipid bilayer into the ICF. Because it can do that, they discovered that target cells will contain receptors for steroid hormones in their intracellular environment inside. So we call them intracellular receptors. Now, we can give them a more specific name depending on whether they're located in the cytosol or nucleus. But in this case, this is an intracellular receptor known as the cytosolic receptor. And what will happen is it will perform reception in the cytosol or in the ICF. It won't perform it facing the ECF. It'll perform it inside of the cell. But reception happens here. And if you notice here, they're showing you this receptor, there's nothing ever linked to it. So we'll talk about this a little bit more when we talk about the second step. So once reception has been performed here at the cell surface or in the intracellular, next is transduction. More specifically, in A&P classes and advanced cell classes, we refer to it as signal transduction. In other words, the signal received or the message received by the receptor has to be relayed to the correct area within the cell so that the cell can perform step 3, the response. The question is whether the receptor that performs step 1 is a cell surface receptor or whether it's an intracellular receptor. Okay, so I'm going to visit this in a sec, but I'm going to go back to this other one. This second step, transduction, I'm going to talk about intracellular receptor first, okay, because it's a little quicker. When an intracellular receptor performs the first step in this 3-step process of a target cell of reception binding to its specific ligand, notice what they show the change in this intracellular receptor. Going from here to here, not only is it binding to the ligand or steroid hormone testosterone, but they put a little yellow halo around it. This is to indicate that this intracellular receptor has become activated. A change has occurred in it due to it binding to its steroid hormone. And what has been found for intracellular receptors, when they bind to the steroid hormone that they're specific to, it causes this intracellular receptor not only to become active, But these receptors start to behave like what we call transcription factors, which is right here that I'm underlining. Transcription factors are proteins, okay, that help a cell read genetic information stored in DNA in order to make a protein to do work. So what will happen is these intracellular receptors, when they bind to the steroid hormone or ligand they're specific to, that'll cause them to become active and move in the direction of where DNA is, along with other proteins, help perform a process that we call gene expression, which results in the production of new protein. Once this new protein is produced, now the cell can carry out the response it needs to do. However, for cell surface receptors, it's not that easy. These receptors are transmembrane proteins. They are part of the membrane. They're not going anywhere. So how can a cell surface receptor that performs reception let the rest of the cell know, hey, we received a signal and we need to respond to this signal? Well, the way cell surface receptors relay their signal to the correct area of the target cell so it can respond is sometimes, not always, as I said, they can focus on ligand-gated or function as ligand-gated channels or enzymes, but sometimes these cell surface receptors will be connected to one of many molecules and ions that collectively together, up here in the top right, that collectively together form a network of molecules and ions that we call the second messenger system. Okay? So listen carefully. There is a network of molecules that are linked to each other and that are linked to the receptor or associated with the receptor. All of these molecules, and I left off ions, but it includes ions, form a system called the second messenger system. This second messenger system creates a pathway known as the signal transduction pathway. In other words, this second messenger system will perform the second process called signal transduction. So for cell surface receptors, the second step in a target cell happens in the ICF. It involves a network of proteins that form the second messenger system. And the job of this network of molecules and ions is to relay the signal received here by the cell surface receptor to the correct area within the cell in the ICF so it can respond. Okay, so you can see here, what they're not showing you is that when this cell surface receptor bound to its ligand, the ligand being the first messenger, this causes a change in the receptor. It causes it to become active. Okay, and if it's a G protein coupled receptor, the G protein is in reference to the first of several molecules and ions that make up the second messenger system. Okay, this is one of the, if not the most popular example of a molecule that's part of a second messenger system. But what happens now is the receptor will tell this molecule here, G protein, I received a signal. This is what it said. I need you to relay it to the correct area of the cell. Then the G protein, it's like a relay mechanism. We'll relay it to another protein, to other molecules. It may involve ions helping to relay that signal till it reaches the correct area in the cell so it can respond. So everywhere where you see the black line, that is signal transduction. And then in between these black arrows or black lines are the molecules and ions that make up the second messenger system that perform signal transduction. I'll let you look at these slides. It's just giving you examples of molecules and ions that can form the second messenger system and play a role in signal transduction. This slide here I put at the top. It's additional information, not on the next exam, but I started putting it in because I had a lot of students going into A&P, and I just wanted them to have an idea of at least be familiar with this. But I'm not going to go over it because it won't be on the exam. It's just extra for you. Okay, step 3. Okay, is the response. Well, the catch with the response is it depends on the target cell. Okay, if it's an intracellular receptor, okay, as I said before, these receptors, they're not stuck in the membrane. They have the ability to move around freely, okay, within the cell or inside of the cell. Okay, so when they bind to their steroid hormone, their ligand called a steroid hormone, that activates them, and if they're in the cytosol, they'll move into the nucleus. If they're already in the nucleus, then they're still going to move in the same direction as this receptor that's being shown here. They will move towards DNA. They will help stimulate the production of proteins. So the receptor itself, the intracellular receptor itself, carries out transduction. What they're not showing you is that the protein will carry out the response. Okay, it's showing you sometimes cell surface receptor through its second messenger system could also end up resulting in protein being produced to get the response out. No, this is not inside a ribosome, Francisco. This right here is the ICF. This is the cell membrane up here. And then over here, this is a better figure. This is a nuclear envelope, the membrane of the nucleus. This is the cell membrane and the cytosol. Now, if you're asking me about this here, okay, this is showing you the nuclear envelope. Notice it's showing you nucleus and DNA. So we're not talking about ribosomes at all. Does that help you out? Francisco, did that help? Yeah, but yeah, out here I show them to you. But we don't need to go into details with these guys yet. Okay. You're getting ahead. That's good, Francisco. I like that. I'll give you ribosome information later, though. Okay. So, all right. Okay. So if you look here, many factors determine a target cell's response. Well, if you have a job, you might be familiar with this. Okay. You might have one supervisor. Okay, telling you what to do and telling you to do one thing, so you respond in one way. Sometimes you have a supervisor telling you to do something, okay, but you realize that something he's actually telling you to do is going to cause you to have to perform two responses, okay? So what they're showing you here is sometimes when a signaling molecule binds to just a cell surface receptor, the cell surface receptor will be connected to a second messenger system, okay, that transduces the signal, resulting in just one response. Sometimes a cell surface receptor will bind to a ligand that will transmit or transduce its signal to the second messenger system, but then the system gets relayed into two separate, goes from one second messenger system to being relayed into two separate second messenger systems to ensure the cell responds two ways. Now this next one you may really be familiar with. This is where you have more than one supervisor telling you to do the same damn thing. Okay, yes, I use damn because I'm thinking about I have so many I lost count. Okay, in this case, you could have two different signaling cells using two different ligands that bind to two different cell surface receptors, but on the same cell. But what happens is both of these receptors are connected to second messenger systems that eventually relay their signal into the same direction, resulting in just one response. And then sometimes, if you look here to the left and to the right, sometimes you have a signaling molecule that can bind to more than one type of receptor, because what the receptors have in common is their ligand binding sites have the same shape. But the receptors overall have a different shape and are connected to a different second messenger system. Therefore, we get a different response. So it is very complex, a very complex process. But everybody, if you've taken medication or you have allergies and you take your prescription meds, okay, what you're taking are ligands to elicit a response from cells of your body that aren't making you feel too good at the moment. So cell communications are very important in the pharmaceutical industry. Now, and to your doctors in your health field. Okay, so now we're continuing with responses, and I believe this is around the last page of your notes. I give you 4 examples of basically how complicated the response can be, or how the response can vary is probably better. Okay, for the endocrine system, whenever you go over the endocrine system, the star endocrine gland is known as the pituitary gland. It's located at the base of your brain. Okay, and it communicates with other endocrine glands such as the thyroid gland. Okay, thyroid gland roughly located around the front of your throat, for a guy around the Adam's apple roughly. Okay, and pituitary gland is located at the base of your brain. The pituitary gland contains cells that produce a hormone, peptide hormone, TSH. TSH, thyroid stimulating hormone. Okay, TSH, okay, in its name, is the target. It targets the thyroid gland, and it's going to stimulate the thyroid gland, and then the fact that it has hormone lets you know it's a ligand or a chemical signal. Well, when TSH reaches the thyroid gland through your blood, okay, it doesn't stimulate every cell of the thyroid gland. It only stimulates those cells of the thyroid gland called follicle cells. They're the only cells, and it says follicular down here, they're the only cells that contain receptors. Okay, these follicle cells are the only cells that contain receptors that recognize and bind to TSH. And the response of your follicle cells, which is here, of your thyroid gland, is to respond by releasing its own hormones. They're called thyroid hormones in lay term, but those thyroid hormones are known as T3 and T4 among the scientific community. Okay, so if you take AMP or if you've had it and you've talked about T3, T4, you know these are ligands referred to as thyroid hormones. So the response by the follicle cells is pretty simple. Hey, I received a chemical signal in the form of a ligand called TSH, and I responded by producing my own chemical signals, ligands called thyroid hormones. Now, sometimes a cell can respond to more than one ligand because they have more than one type of receptor. In this case, I'm going to use your heart, specifically the myocardium of the heart. Everybody, myocardium is a fancy way of saying cardiac muscle tissue, and cardiac muscle tissue is made up of cardiac muscle cells. Some of these cardiac muscle cells have receptors, okay? Two different types of receptors that recognize two different types of ligands. Some of these cardiac muscle cells, I'm just saying some, will have receptors that recognize a ligand known as ACH. Down here at the bottom, ACH stands for acetylcholine. Okay, it functions as a neurotransmitter in this case. These cardiac muscle cells also have receptors that recognize a different ligand. It's called norepinephrine. The abbreviation for norepinephrine is NE, and as I told you before, norepinephrine binds to a class of receptors known as adrenergic receptors. Now, so these individual types of cells, okay, how they respond depends on which of these ligands interacts with which receptor. The way it works for norepinephrine, okay, when the nervous system wants your heart rate to go up, okay, pump blood faster, your nervous system will release a ligand called norepinephrine. Norepinephrine, like I said, you can think of it as a cousin or sibling of epinephrine. Therefore, it binds to the same class of receptors known as adrenergic receptors. You do not need to know the Greek letters. I just put them there so you know what you're looking at over here. Okay? When the nervous system releases norepinephrine, okay, and it binds to the adrenergic receptors on some cardiac muscle cells, what norepinephrine does is it positively stimulates the cardiac muscle cells to increase your heart rate, HR, which I put over here. Okay? So norepinephrine, in this case, is a ligand that's functioning like the gas pedal. Okay? It's saying speed things up. This other ligand called acetylcholine targets the same type of cell, cardiac muscle cells, except it binds to a different class of receptors known as acetylcholine receptors. Now, there's more than one type of acetylcholine receptor, but on cardiac muscle cells, the acetylcholine receptors are known as MACH receptors or muscarinic acetylcholine receptors. In this case, when the nervous system wants your heart rate to slow down, your nervous system will release a ligand called acetylcholine, ACH. It will bind to these types of receptors known as muscarinic acetylcholine receptors, and that will have a negative effect or an inhibitory effect on your cardiac muscle cells. And if you look over here down to the bottom right, acetylcholine is associated with decreasing your heart rate. I'm sorry, everybody. Decreasing your heart rate. So this acts like the brake. Okay. The only thing we don't ever want happening in regards to your heart, obviously, is it completely stopping. Okay. But Our nervous system is responsible for telling our cardiac muscle tissue or our myocardium when to speed it up or slow it down. Now, sometimes the response of the cell, okay, or sometimes, I'm sorry, a one type of ligand can actually elicit different responses from different types of cells, okay? So that ligand I talked about, acetylcholine, okay? Right here, I'm sorry, over here to the right. This ligand acetylcholine. Well, what they found out is this ligand acetylcholine not only binds to receptors known as muscarinic acetylcholine receptors, they found there was another class of acetylcholine receptors called nicotinic acetylcholine receptors. Listen carefully. I already talked about the muscarinic acetylcholine receptors. You find them in the membranes of cardiac muscle cells. That's one way. And it has acetylcholine has an inhibitory effect. I don't know if I'm going to make it here. However, on skeletal muscle cells, this ligand acetylcholine has a complete opposite effect. Okay? On skeletal muscle cells is where you will find acetylcholine receptors known as nicotinic or NACH receptors. And when acetylcholine binds to these nicotinic acetylcholine receptors on skeletal muscle cells, it actually stimulates the skeletal muscle cells to contract and produce movement. Okay, this is important in regards to if you're familiar with Lou Gehrig's disease, also known as ALS. Okay, if you see a person with Lou Gehrig's disease, what you see is what we call muscle atrophy. The muscles are wasting away. Okay, usually from the time of diagnosis, the person roughly has about 5 years to live. Treatment is getting better. But what you see is muscle atrophy. But in actuality, it's a neurodegenerative disease. In this case, when it comes to your skeletal muscle cells, it is your nervous system again, just like with cardiac muscle cells, it is your nervous system again, okay, releasing this ligand called acetylcholine, telling your skeletal muscle cells to contract. But in Lou Gehrig's disease, the cells responsible for producing acetylcholine that will stimulate your skeletal muscle cells to contract, those neurons in the nervous system are dying off. So what happens is the skeletal muscle receptors, these nicotinic acetylcholine receptors, they never interact. They're never able to perform reception because there's no acetylcholine. So what happens? Your skeletal muscles start to atrophy or waste away from inactivity. So this is another area, okay, that another reason why cell communication is so important. If you look here, another way, the fourth way, the type of signal transduction pathway can also determine what type of response we have. So here, if you look down here in the white box, we have the same target cell. It's a smooth muscle cell. Okay, both types of target cells have adrenergic receptors. Now I said, don't worry about the Greek symbols right now. I'm cheating a little bit. They both have adrenergic receptors. And never more epinephrine. Look at epinephrine. It's showing you how epinephrine can bind to receptors on these target cells over here that are smooth muscle cells, or it can bind to these receptors on these smooth muscle cells here. Now, it depends on where the smooth muscle cells are located. Okay, you'll find smooth muscle cells, two popular areas, in the walls of your blood vessels and in the walls of your respiratory tract, your airway. Okay. Now, so we have the same ligand, we have the same class of receptors, yet over here to the left, it says the response by these target cells, by these smooth muscle cells, will be to relax. Over here, it says the response by these smooth muscle cells, these target cells, will be to contract. Why? Are generally the same and the ligand is the same. It has to do with step 2. The network of molecules and ions that make up the second messenger system are different. So that can cause a cell to respond differently. So you can have two muscle cells but located in two different regions of the body. They both have the same class of receptors. They both recognize the same ligand, but the second messenger systems are different. Therefore, step 2, signal transduction, okay, is a little different, ensuring that we get different responses. So it is a complicated process. So this right here on this side that I'm circling, this is trying to represent how busy it is inside of a cell. This is a cytosol. This is inside the nucleus. Okay, so you have to envision the activity that's going on inside of a cell in the intracellular environment is like a busy city. There's a lot of activity going on, and then to make it even busier are all the chemical signals, the ligands, okay, that are binding to receptors in the cells, further increasing the amount of activity in a cell. So it is possible, okay, that something can go wrong in the cell. Our cells have a safeguard built into them, okay, to where either they can only live for so long, or if something goes so wrong it can't be fixed, they will go into a process called apoptosis. Now, it looks like apoptosis, but it's apoptosis, okay? But if you spell it right, I'm happy. Apoptosis. Some people call it cell suicide, but I have that in your notes just in case you hear that. Really, apoptosis is defined as programmed cell death, and I have it down here too, okay? So excuse the use of this right here, but that's what I started hearing some professors and some students say, and I just want to make sure when you see this, okay? Yes, self-destruct, like self-destruct, exactly. You'll see when something self-destructs, except in this case, when self-destruction occurs, we want to make sure it's controlled, organized, so that neighboring cells are not damaged, so that neighboring tissue is not damaged, okay? So if you look here, this is a normal white blood cell. But white blood cells have, you know, their jobs aren't the nicest. It's like being in law enforcement, okay? The job of white blood cell is to go where there is trouble. You know, I have a son that's in law enforcement, and I tell him his job's tough because it is his job to go to where there's problems. Okay, so it's a dangerous job, but sometimes something can go wrong for the white blood cell to where it has to undergo apoptosis. So this figure here is just showing you, it's showing you live pics, but it's showing you a simple diagram. When something goes so wrong with the cell that it can't, how do you say, reestablish homeostasis, and it's going to have to undergo apoptosis, either the cell itself will recognize it, okay, and you'll have an internal signal occur, or a neighboring cell will come up and say, I notice you're not functioning properly. We need to have you remove yourself. It sounds cold-hearted, but it happens in our body. So in this case, imagine this cell. Something has gone so wrong, it has to undergo apoptosis or programmed cell death. There's a class of enzymes called Caspases that will be produced, different types. And the job of these enzymes initially is chop up everything, cut it up. Whether it's a non-membranous or a membranous organelle or just compounds, chop them all up, break them all down. Then what will happen to the cell undergoing apoptosis is it'll start to break apart into small vesicles that they call blebs. Okay? So in other words, the plasma membrane starts pinching off, drawing in some of the intracellular environment containing all the degraded compounds and organelles into these small lipid packages known as vesicles, but they call them blebs. Okay? This is to ensure that any compounds, even though they've been degraded or destroyed, this is to ensure that they're not released to where they can come in contact with neighboring cells and cause any damage. We want this cell to remove itself without hurting anything else. So it'll break apart into vesicles called blebs, and then white blood cells capable of phagocytosis, sometimes called phagocytic cells, will come up and endocytose these blebs, clean up the area where the cell, okay, that died was, as if nothing was there. Sounds a little cold-hearted, huh? So it happens in our body, everybody, okay? So I know it's a lot. So like I said, make sure you use the notes, the PowerPoint, go through the review, but also read your text for sure, okay? Yeah, it's a rough life at the cellular level all the way up to the macroscopic level. So listen carefully. Are there any other questions? As far as lab goes, we will address lab questions in lab, but do read that document I sent out on Friday carefully, please. Carefully. I even numbered it, first, second, third, and fourth. And if you go to third, it answers the questions because I got a lot of questions about pre-lab, okay? There's another review for Chapter 11 if that's what you're asking me. Okay, hold on. Thank you for reminding me. I actually did upload it. Okay, here we go. I got it right here too, LaCour. So here's the review. Okay, so here's the review. Again, I ask you some questions on the second, and then there are a few diagrams. Oh, I'm glad you did bring it up, LaCour. Thank you. On these diagrams that you have to label, I tried to give you a little more information so you have an idea of what to label, because Chapter 11 is a technical chapter. I'm fully aware of that, okay? All right, so what I'm going to do, everybody, is if there are no questions in relation to the lecture, okay, I'm going to have y'all leave the session, and then I'm going to start a new session, which I already have posted so you can see it. I'm going to start a new session at 1:30, and I'll go over lab, and that's when you can ask me any additional questions, okay? Listen, online's hard, and I say that on the video that I posted on Sunday. Unfortunately, that video I posted on Sunday led to more problems for me. So trying to do more, I keep forgetting what I was taught. Less is more, and unfortunately, I did more, and the results are not what I wanted. They're a lot less. But listen, okay, just stay on top of things, and you're going to have to put more emphasis on details when you read something, okay? I'm trying to. I'm trying to. Okay, if there are no more questions, please go ahead and leave the session. Don't forget, Francisco, you have to leave the session. Okay. And I'll see the rest of y'all